Today we're going to be discussing inflammation. And this is a topic that's going to be covering a lot of different aspects of the immune system that we've discussed in previous lectures. There's also no specific chapter in the book for this talk, but I'll be drawing with some figures from chapter 3, as well as from some current research papers, which I will reference throughout the talk. So today we're going to start out discussing what is inflammation. And then we'll talk about the canonical inflammatory response that is most often discussed, which is the inflammatory response to infection. From there, we'll discuss some inflammatory mediators, some important molecules that help aid with the inflammatory response. And we'll discuss the differences between acute and chronic inflammation, as well as a few examples of diseases. And when we discuss diseases, it's not important that you guys memorize each disease and the symptoms, things like that. It's more so that I want to use these diseases as a framework in which to discuss inflammation in general. So we'll start out with what is inflammation. So one of the common definitions of inflammation is an activated immune state in response to infection, injury, or stress. And these three different categories are all very important. Like I mentioned, infection is the most common situation in which we discuss inflammation, but we'll talk about a few examples from injury or stress and how the inflammatory response aids in restoring those tissues. So it's possible that inflammation may have evolved not to fight off pathogens, but actually to return the system to homeostasis. So what this means is that the tissue has a baseline. If something happens, like an injury, whether from an impact or cell death, even if there's no infection there, it's really important that the system is able to return to that original homeostatic state. It's also typically thought of as the initial response to infection or injury. So the inflammatory response is going to be involving a lot of the innate immune system. And it's really important that we think about the inflammatory response and how it brings different immune cells and different immune mediators to that site of infection or injury. It's often a nonspecific response as well. So we'll discuss this a little bit, but as you guys talked about with the innate immune system, this response is not specific. So the response is mounted against any pathogen, injury, damage, and it leads to a large inflammatory response and influx of cells, and those cells can cause off-target effects such as additional tissue damage. So another part of inflammation is actually the resolution and repair stage. So besides mounting a response, the body also must return back to that homeostatic state. So we'll first start out talking about the canonical inflammatory response. And this is the most well-studied response, and it's typically to infection. And here, this is from chapter 3. You guys will have seen this when you discussed the innate immune system, but this is an example of the initial response. So if we have a microbe, the macrophages or other innate cells will come in, sense the microbe, and release inflammatory cytokines. Those cytokines go into the blood vessels and initiate a set of reactions which allow circulating leukocytes to know where to enter the tissue. So this is all initiated by recognition of PAMPs or DAMPs, which are your pathogen-associated molecular patterns or damage-associated molecular patterns. And cytokines and chemokines released by these first cells are really important for mediating leukocyte migration and that immune response to that location. So once the endothelium here sees those cytokines, it will upregulate molecules on their surface so that leukocytes in the blood will come and stick to the endothelium, and so they can pass through from the blood into the tissue. And this inflammatory response also initiates a micro blood clotting response as well. So here's an example of some details of that extravasation process. So here we can see the immune cells rolling along the endothelium here in blue, and they will interact with different adhesion molecules on the surface of the endothelium. Then once they stick to one area, they can pass through the endothelium into the tissue of interest. And this is really important because these cells go specifically to that area. They're brought in by the release of cytokines and chemokines. And so this is how the cells know where to go. And they will also then, once they get into the tissue, differentiate into different cell types as well. So monocytes might go in and differentiate into macrophages, for example. Another really common cell type that undergoes this process is the neutrophil. And we won't be going into details about that since it was discussed earlier in the innate immunity lecture. But just know that neutrophils in particular have off-target effects. So they come into the site of inflammation where there's been a release of inflammatory cytokines and chemokines, and they'll release granules and undergo reactive oxygen species release as well. And this can cause damage both to a pathogen as well as to any healthy tissue around it. It's not specific for a pathogen. So this response leads to an influx of components of the immune system. So we discussed some of the cell types like neutrophils and leukocytes, but this also allows the influx of components from the blood, such as blood plasma and antibodies. And this allows the immune system to access those tissues that normally are behind the barrier of the endothelium and basement membrane. And the goal of this inflammatory response is to allow the immune system to return the tissue to homeostasis. And that involves finding the pathogen, removing the pathogen, and then cleaning up any damage that was left in the tissue and restoring that tissue back to its healthy state. And immune cells, like I mentioned, can release mediators that eliminate the pathogen, but they can also cause indiscriminate damage. And so it's important to eliminate the pathogen, but it's also really important to remember that these responses are followed by that resolution and repair stage. And this stage is often mediated by macrophages, especially tissue-resident macrophages, which can sense damage and help repair the tissues. So this is the typical inflammatory state, and it's the most researched state of inflammation, so it's probably something you guys are familiar with from earlier lectures. However, inflammation is much more than just a response to infection, and we'll be discussing some of those examples as well. And this is a topic of current ongoing research, so I'll be referring to some recent papers, but keep in mind that this is all fairly new and new information is coming out all the time. So I encourage you guys to do a bit to do that because inflammation is such a broad topic. We'll be touching on some of the highlights, but it's impossible to cover this entire topic in just a 45-minute lecture. So we'll be talking now about inflammation beyond infection. And so the inflammatory response can also be a response to sterile tissue injury or to an altered homeostatic state variable. So what does this mean? So a variable is something that is sensed by the body, and it's usually a molecule such as glucose levels that needs to be kept in a very narrow range. So inflammation can help keep that variable within the healthy range, and we'll talk about that a bit more. So inflammation can be induced by many different things. We mentioned pathogens, so that's an exogenous source, which is sensed through TLRs or pattern recognition receptors. But there are also some endogenous sources of inflammatory signals. So this includes signals that indicate damage. So for example, if there are components of the extracellular matrix that are seen, these normally in a healthy tissue would not be sensed. But if there's tissue damage and breakdown of the ECM, these will be released, and cells in the body can sense them as being out of place. It's the same for nucleic acids. So typically DNA and RNA are never found outside the cell. So if this is sensed in an area outside the cell, that can indicate to the immune surveilling cells like macrophages that there has been cell death, and that is why this is released. So those are some of the initiating signals. So I mentioned some variables and returning those variables to homeostasis. So this is an engineering-based diagram to help you guys visualize what that means. So we have a value, for example, the blood glucose level, and this is sensed by various cells in the body, for example, macrophages. Then this is sensed by a cell or a controller that provides a signal to the plant or to the other cell type that's able to alter that variable. So in this case, this might send signals to the liver to release glucose, or to the pancreas to increase the release of insulin. So this then feeds back and changes the value of that regulated variable. So the release of insulin will return the blood glucose down to the value that it needs to be in for healthy tissue function. And you can see that example here. So again, we have our blood glucose. The pancreas senses this and can release insulin to, for example, skeletal muscle. And the skeletal muscle can then uptake glucose and use it, and this will reduce the blood glucose level, which again is sensed by the pancreas. So this homeostatic circuit helps explain and visualize what's going on in the body. So an inflammatory response in this context would be the response that is mounted to return that variable back to its set point. So I mentioned there are a couple different instigators of inflammation, and all of these each lead to a different physiological response and can have different pathologic outcomes when the homeostasis of this system is not maintained. So here we have some examples of infection, tissue injury, and tissue stress. So starting with infection, it's the first situation we discussed. So infection induces inflammation, which then induces a host defense against that infection. So if this is dysregulated, you can see things such as autoimmunity or immunodeficiency, so a badly tuned immune response, and it can lead to tissue damage. Similarly, tissue injury without an infection can lead to a tissue repair response. And it's important to note here that this is not necessarily what we would consider a stereotypical immune response. Tissue damage won't typically initiate the influx of neutrophils, inflammatory mediators, unless it's very large tissue damage. So the sensing of tissue injury by macrophages will induce this tissue repair response. However, this can also be dysregulated and lead to pathogenic outcomes such as fibrosis. Finally, we have tissue stress and malfunction. So this would be an example such as a tissue that is in an environment at the wrong temperature or without enough oxygen to function. And this is going to cause some damage. So the inflammatory response here to the tissue stress is going to be to try and adapt to that stress and return to the homeostatic state. And again, this can lead to dysregulated outcomes, and we'll talk about this a bit more when we discuss chronic inflammation, but this can lead to a shift in that homeostatic set point. So with our glucose example, if there's a normal level of glucose that you want to have in the blood, it's possible that if the body cannot change that, it will just change the level that it wants. So this is an example that you'll see in type 2 diabetes, where there's resistance to insulin. So this is just a summary of some of the initiators of an inflammatory response. And here I mentioned exogenous and endogenous inducers. So exogenous would be our microbes or non-microbial things such as allergens or toxins. Endogenous initiators would be such things such as tissue or cell-derived damage signals. Okay, so we've talked about the initiation of inflammation, but now let's discuss some of the ways that this response is mediated. So there are 7 different categories of inflammatory mediators, and we'll go through and see an example of each. And these all help to propagate the immune response. So these mediators will have effects on other immune cells, on the endothelium, for example, to help return that tissue to homeostasis. So our first example are vasoactive amines. And one example of this would be histamine, which we'll discuss in the allergy lecture. And these chemicals help change vascular permeability. And this change allows for an increased influx of immune cells or cytokines into the tissue. We then have vasoactive peptides such as substance P. And these they release those granules and cause an inflammatory response. So we can also include complement fragments in this list of inflammatory mediators. So as you guys discussed previously, complement cascade occurs, and there are many different mediators that are cleaved. And one example is the C5a protein. And C5a, besides being involved in complement, also acts as a chemokine. So that means that it can recruit additional immune cells to the site of complement activation. So for example, this could recruit monocytes from the blood. We then have lipid mediators such as prostaglandins, which we'll go into a bit more detail about these in a second. And then we have our most well-known inflammatory mediators, which would be cytokines such as TNF-alpha. And these can do quite a variety of functions, including activate the endothelium to help your inflammatory cells to bind and exit into the tissue. And they can also help induce an acute response. We also have chemokines, which are similar to cytokines, but they induce migration to the source of the chemokine. So these, again, help control the immune cell infiltration to that area of infection or tissue injury. And finally, we have proteolytic enzymes, such as matrix metalloproteinases. And these are repair for tissue repair response.